Hello class, this is Chapter 5, Part 1. Chapter 5 is over the plasma membrane and movement across that membrane. With a little bit at the end about cell junctions. Anyway, let's get into 5.1. It's the introduction and a little bit about plasma membrane structure. So every cell has a plasma membrane. This includes bacteria, plants, animals, fungi, and protists. So it's common to all cells. It has a few functions. One of it is to separate the inside from the outside. The inside is the cytoplasm or cytosol, and the outside is the environment or maybe tissue fluid in like a body. And its other job is to govern what comes in and out of the cell. It has two major components, and this is consistent with the fluid mosaic model. The fluid part are these phospholipids. And the phospholipids make up most of the membrane, most of the barrier part of the membrane. So the phospholipids are what separate the inside from the outside. When you put phospholipids in water, they'll make a bilayer where they hide the hydrophobic tails. And it's the hydrophilic polar heads that will interact with the environment, with the water on the inside in the cytosol and the water on the outside in the environment. And we'll see a picture of that in a second. The mosaic part is the many different proteins. The proteins are sort of like the doorways and gateways. They allow things to pass through the membrane. Whether they physically pass or it's just the communication that passes. And the membrane is also what's presented to the outside world. So we'll have some features due to that as well. So here's a 3D cartoon of a plasma membrane. These little spheres are the hydrophilic heads of the phospholipids. These two little squigglies coming out of them are the hydrophobic tails. And I want to show you this bilayer structure where the phospholipids are inverted towards each other. So the tails are facing in and the heads are facing out. And that's because the heads are hydrophilic and they can interact and play with water. And there's water inside the cell in the cytosol and water outside the cell in the tissue fluid or the environment. And again, the phospholipids are the flexible part. They are very mobile. You can see this if you've ever blown bubbles. Soap is made out of phospholipids. And if you, before you start blowing a bubble, it's sort of a flat sheet in your wand, and then you exert pressure on it and it bends, and that bending is showing the flexibility of phospholipids in a solution. The membrane needs to be able to change shape in order to function properly, so that's a really important part of the phospholipids. The other important component is this hydrophobic area in the middle. It will block anything that is water soluble. So it will block water and anything water soluble, which includes anything polar and anything ionic. It'll also block things that are large, whether they're hydrophobic or hydrophilic. So those phospholipids make a good barrier, and they're also highly flexible. The other component are the membrane proteins. That's not really easy to see. Let's just do black. And these membrane proteins typically allow transport, either physical transport, they allow cargo to pass through them, or they will physically bring cargo through the membrane. The other way they often function is in signaling, so they will allow a signal to pass from the outside into the inside of the cell. Some other components of the membrane, at least one that's really important, is this guy right here, which from chapter 3 you should recognize as a sterol. At least it is cholesterol, and cholesterol helps the membrane be very stable. So if you heat membranes up, they can disintegrate. Basically, they will dissolve and separate, and that barrier will be lost, and that would be very bad for the cell. That's actually one way we kill bacteria, is by punching holes in their membranes. And so cholesterol helps to hold it together at high temperatures. Also at low temperatures, that membrane becomes rigid and not flexible. And that can be a bad thing as well, because the membrane needs the flexibility to function. And so that cholesterol helps it be a little bit more flexible at lower temperatures. So it helps with both extremes. So it is an important component, but it's not a major component. Not as major as the proteins or the phospholipids. Alright, one of the other minor components are these carbohydrate attachments. They can vary in length and shape and branching, but they're made out of sugars, typically glucose and fructose. And when they're attached to proteins like this guy, we call it a glycoprotein. And when it's attached to a lipid like this guy, we call it a glycolipid. So we just put that prefix glyco to refer to the fact that it's modified with some sort of carbohydrate modification. And these exist, remember when I said the membrane is what is presented to the outside world. A lot of that comes from the glyco modifications. They are typically identifiers like your driver's license or your birth certificate work for you. These identifiers work for your cells. So they'll say like, I'm Dr. Hilton's cell, I'm a liver cell, I'm 23 days old, I'm virus free, I'm cancer free. Those are the types of signals. It says using these glycoproteins. Again, they are just identifiers. They're for identification. Alright, when I was talking about proteins, I forgot one small detail. You can have two types of membrane proteins. If a protein goes through both layers of the phospholipid bilayer, so here's one layer, here's another. The technical term is leaflet, so this would be one leaflet and that would be the other. So if your membrane protein goes through both layers, both leaflets, it is an integral protein, and it's really hard to remove from that membrane. That's one reason we call it integral. The other type, these proteins are anchored to one side or the other. They're loosely anchored and they only go through a small portion of the membrane, or they may just have a fatty part that goes in and anchors them. But these proteins are called peripheral proteins. Peripheral means to the side or around the side, and they are on one side or the other of the plasma membrane. So I think that's all of the major components. Again, know the fluid mosaic model. The mosaic part is just saying the membrane's made up of many parts, and the bulk of that is many different proteins. The more minor parts included in that are cholesterol and the glyco modifications. And then the fluid part is the phospholipids. They make this really great barrier that block anything that is polar or ionic and large. So the only thing that can pass through has to be small and hydrophobic or nonpolar, at least. And so this plasma membrane, these phospholipids block most things from crossing the membrane, and then the proteins will allow transport of things we want. And the phospholipids are very flexible, and that's the fluid part. Okay, so let's get into some functions of these membrane proteins. The main point here is that there's lots of different jobs for these proteins. We'll need to know some of them in detail. One of them you need to know in detail are these channel proteins. These channel proteins are basically a protein that create a hole or a passageway or a tunnel through the plasma membrane. They are specific for their cargo based on size and shape and charge. Related, but you also need to know it, is a carrier protein. And these also transport things through the membrane, but they're a little bit more interactive. So they will actually bind to their cargo, and it'll physically pass this cargo through the membrane, whereas the channel proteins, those are a little bit more passive. Usually these proteins, either carrier or channel, are dependent on diffusion. So you need a concentration gradient. So you'll notice there's lots more cargo out here and very little cargo in here. So the cargo will want to travel in to balance out. And once it's balanced, the transport would stop. So know your difference between channel and carrier. Again, channel proteins are really passive. They are holes in the membrane or tunnels in the membrane that allow passage. Carrier proteins are a little bit more interactive. They will physically grab onto their cargo and move it across the membrane. All right, cell recognition proteins, those are those identifiers I talked about, hence the glycomodification. And they're just like a driver's license or something like that. Receptor proteins, these are for communication. So instead of taking cargo and passing it through, anytime it binds to its partner, it'll cause some change on this part of the protein, and then that signal will be moved into the cell. So it's kind of like the game Telephone, where someone tells you something, you tell someone else something, you tell someone else something. That's how receptors work. So this is the first messenger. It fits like a lock and key with the receptor due to that shape and charge. And when they fit together, that'll activate this receptor and it'll talk to the inside of the cell. The telephone analogy breaks down because usually in telephone you lose information. These receptor proteins are very good at maintaining information. So receptors may be a little bit hard to understand. So let's think a little bit about capsaicin. Capsaicin is sort of the active ingredient in a pepper, like a jalapeno or habanero, that tells your body it's spicy. It'll cause some pain and some sweating and some other things related to hot or spicy. Capsaicin binds to your cells at the plasma membrane. It specifically binds to a membrane protein. This membrane protein is on a nervous cell, a nerve cell, that senses pain. When capsaicin binds to this protein, that protein causes channels to open, and then calcium flows into the cell. Calcium, remember, is an ion. It's usually got a +2 charge. And it's commonly used as a signal in cell signaling, in cell communication. So the calcium enters the cell and that causes the nerve to fire, and it'll send signals to the brain. So capsaicin will bind to our receptor. The receptor will open and cause calcium to enter our cell. And then when calcium's in the cell, then the nerve fires and it'll send a signal to the brain. Some membrane proteins are enzymatic, but enzymes are in the next chapter, so I'm not going to worry about that too much. And some membrane proteins are for connecting cells to each other. So here's the plasma membrane of one cell, here's the plasma membrane of another, and they're connected by this membrane protein. They each have a copy and it binds to itself. Besides cell-to-cell attachment, there's also membrane proteins to attach to the extracellular matrix. The extracellular matrix in your body is often a mixture of fluid and a network of proteins, or a scaffolding of proteins. This is just a presentation of all of those, but I think it was easier to go through one at a time. Alright, so let's look at cell signaling a little bit more. Cells have to talk to each other. That's how your nervous system controls most of your body. And it's how development happens. It's how we go from an egg to a person. And it's very important for homeostasis. You know, when you're like craving some specific food. My nephew used to crave cucumbers. That's probably due to cell-to-cell communication, saying you're deficient on something. So it starts with a receptor. So here's our receptor molecule, and here's its partner, the signal. The signal is also called a ligand. And that word ligand means to bind. And it binds to our receptor. And each ligand has a different receptor, or you could flip that. Each receptor has a different ligand. They fit together like a lock and key. Anyway, when they unite, when they bind together, and you know, binding is kind of a weird concept too, but you know, imagine giving someone a handshake. I know we're not doing that very often now, but when you give someone a handshake, you're binding to them. When you give someone a hug, you're binding to them. These molecules, when they bind, they physically touch, and they usually stick for at least some time. Anyway, when our ligand binds to our receptor, some magic happens, and you get a transduction pathway. This is the telephone part, or it's a continuation of the telephone part. So the signal hit our receptor. That's the person starting the telephone game. And then the receptor talks to something else, the little square here, which talks to something else, the triangle, which talks to something else, the sphere. And so passing down that signal, that's the transduction pathway. And then one of those signals will typically go to the nucleus. It'll turn on a gene, and that gene will make some sort of protein. And then that protein will result in a response. So think about insulin. Insulin is a hormone, which is a chemical signal, a chemical messenger, much like this guy. Let me change colors, much like this guy. It is basically the dinner bell for cells. So after you've had a meal, your blood glucose goes up. Your pancreas senses this, and it'll release insulin. Insulin again goes out and tells all your cells they can eat. They can absorb glucose from the blood. They're allowed to. But a few things have to happen for that communication to occur. So the insulin has to go and bind to an insulin receptor on the cell. That signal is transduced or passed through a transduction pathway. That'll go to the nucleus, where it turns on a gene. This gene is for a glucose transporter. So it'll have instructions to build a glucose transporter. That transporter will go to the plasma membrane, and then glucose can enter our cell. And that's how cells eat. So it's all through cell communication, not just between cells, but from the surface of the cell to the nucleus. So one of the main features of the plasma membrane is that it is selectively permeable, or semi-permeable. So semi or selectively means some things but not others, and permeable means passing, or it means it's permissible. So some things are allowed to pass, some things are not. As a matter of fact, it's very specific what it allows to pass. Most things cannot. So anything, well, let's look at the picture. So again, there's water outside of our cell in the tissue fluid or the environment. There's water inside the cell in the cytosol, which is a big component of the cytoplasm. Our hydrophilic, water-loving heads are interacting with both sides of the water. Our water-fearing, I can talk, hydrophobic tails are hiding away from the water in the interior. And they will block anything that is water soluble. So water, ions, and things that are polar, as well as things that are large. So first they're showing you what happens with charged molecules and charged ions. Charged ions are very small, they're one atom, and they are deflected by the membrane, specifically this nonpolar hydrophobic core, the hydrophobic tails. Some ions and some cargo that are small, like water, they can pass through transport proteins. This is a channel protein here. The one for water is called aquaporin. Which is pretty much spelled how it said, aquaporin. And it is a channel protein that allows water to move freely between the inside and outside of the cell. Some charged ions can also pass through a channel protein or a carrier protein, but usually the timing is controlled. It's usually not always open, but there are cases where they're always open for certain ions. And then relatively small non-charged molecules can typically freely pass, depending on concentration and diffusion. So cholesterol is an example of something small and not charged. And then remember how we learned about triglycerides and they had 3 fatty acids attached? Well, if you get it down to a monoglyceride, so only 1 fatty acid is attached, typically this is done through the digestion activity of your digestive system. The monoglycerides can move in and out of the cell pretty freely. as well as short fatty acids. So these are all examples of small non-charged molecules, and we'll get to another example in a minute. And then large things are typically deflected just due to their size. So small and polar can usually go through if there's a membrane protein that is open to them. But if there's no membrane protein, there's no transport of water or ions. The only thing that could pass is something small and uncharged. Alright, so that's pretty much it for part one. We'll start getting into the different modes of transport in part two. There's basically two types. You can have passive transport or active transport. And you'll need to know the difference between those. Passive transport typically follows diffusion as the driving force. So diffusion will determine the direction of movement. And with these passive modes of transport, being as it follows diffusion, diffusion is movement of things from a high concentration to a low concentration, which makes sense that it is downhill somewhat. If you took a ball to the top of a hill and let go of it, it would roll down the hill. Same thing with atoms. If you let atoms do their own thing, they will diffuse from high concentrations to low concentrations. So things will move from a place where there's a lot of them to a place where there's very few of them. It'd be like us moving from the city to the country. So that's passive transport. This typically does not require energy. And again, it's going to be from high to low concentration. Active transport is the opposite. We're going to go from low to high. There's my eye. Which is uphill. And moving things uphill takes energy. And so that's active transport. So that's it for this section. We'll get into details of both of these 5.2s next. It's over the three types of passive transport.